Hello, welcome to biopsychology, the biological basis of behavior. So we're going to start by looking at the nervous system, the central and peripheral nervous system. So when they talk about the nervous system, the central nervous system is the brainstem and spinal cord, running all the way down your spine, up to your brainstem. So this is the kind of control center, processes information and coordinates responses. Going out from the central nervous system like a kind of web everywhere else is the peripheral nervous system. And this connects it to the rest of the body. The peripheral nervous system is then divided into the somatic nervous system and the autonomic nervous system. So the somatic nervous system controls voluntary movements via skeletal muscles and processes information, and the autonomic nervous system regulates involuntary functions and has two branches to that, which is the sympathetic branch and the parasympathetic branch. So this is linked with, this bit here, the ANS, is linked with fight, flight, or freeze. So the sympathetic branch would be activating fight or flight response, and the parasympathetic branch would be promoting rest and digest. So just to reiterate, so the central nervous system is a brainstem and spinal cord. The peripheral nervous system is like a web that goes out from there to everywhere else. And the peripheral nervous system is split to the somatic and the autonomic nervous system. In terms of neurons, three main ones you need to know really: sensory neurons, relay or interconnecting neurons, and motor neurons. So a sensory neuron transmits information from sensory receptors. They have a certain structure, cell bodies offset from the main axon, creating a pseudo-unipolar appearance. The dendrites connect to sensory receptors in the skin, the muscles, and organs, while their axon terminals form synapses with interneurons or motor neurons in the spinal cord or brain. The relay neurons, mainly found in the central nervous system, these connect sensory and motor neurons, forming a complex neural network, and they process and integrate information from multiple different sources. Most neurons in the brain are interneurons, and that allows for complex processing that underlies cognition, memory, and emotion. And they typically have shorter axons than sensory or motor neurons. Motor neurons, they transmit from the central nervous system to effector organs like muscles and glands. They have a multipolar structure with numerous dendrites receiving signals from interneurons or sensory neurons. They have long axons extended from the central nervous system to the peripheral effectors, enabling for rapid transmission of action potentials that can trigger muscles to contract or glandular secretions. So, for example, if I put my hand on a hot plate, the sensory neuron would fire a signal up my arm through the peripheral nervous system up to the central nervous system. It would then interconnect to a neuron in the brain, back down again to a motor neuron to tell me to move my hand. So that's how you'd all three would be sort of connected. When we look at synaptic transmission, so once an action potential is fired down the axon, it will jump the gaps, which is the gap between the myelin sheath, and that's called the node of Ranvier. And it will jump that gap to speed up transmission. When it gets to the terminal button, which is where the vesicles are, it will stimulate those vesicles to move forward, and those vesicles will then fuse with the membrane and release the neurotransmitters across the synaptic gap. Now, synaptic transmission can only go one way because there's no receptor sites on the presynaptic neuron. So once the neurotransmitters, which are the chemical messengers, are released across the synaptic gap or cleft, they will then bind to the postsynaptic neuron, the receptor sites. Now, depending on what they are, depends on whether the neuron will fire or not. So you can get what's called excitatory postsynaptic potentials or inhibitory postsynaptic potentials. So, for example, here we've got glutamate. So, glutamate would increase the chance of a neuron firing, whereas an inhibitory neurotransmitter like GABA would decrease the chance of the neuron firing. So, some of the common neurotransmitters are acetylcholine, involved in muscle movement, dopamine, which is well known for rewards, serotonin, uh, anxiety, depression, mood, and noradrenaline for fight or flight. So, for example, if someone suffers from anxiety or depression, they might be given an SSRI, which is a selective serotonin reuptake inhibitor. So, what this means is that when because they've got a lack of serotonin in the synapse, rather than some serotonin going back through reuptake channels, those reuptake channels are blocked, which means there's more serotonin in the gap, which means it's more likely going to fire the postsynaptic neuron. So selective serotonin reuptake inhibitors means that it's blocking the reuptake. The endocrine system deals with the glands and the hormones. So the master gland is the pituitary gland, and you've got the adrenal glands above your kidneys, thyroid gland, and pancreas. So when you look at fight, flight, you look at some of these glands and how they work. So if you think about the HPA axis, the hypothalamic pituitary adrenal axis, which is used for chronic stress, the hypothalamus would release cortico-releasing factor. That would then stimulate the pituitary gland, which would then release ACTH, adrenocorticotropic hormone, which would then stimulate the adrenal gland to release cortisol. So the HPA axis is for chronic stress, long-term stress, which is obviously bad for your immune system because it can weaken it and maybe result in global immunosuppression. Whereas if you look at fight-flight response, this is the SAM pathway, short-term stress. So the SAM pathway is a sympathetic branch and the adrenal medulla. So when you perceive a threat, the amygdala is activated. That kind of triggers off the danger response. The hypothalamus activates the two branches of the autonomic nervous system. The sympathetic branch would start then releasing adrenaline. It gives you all the physiological changes to adrenaline, which are here. Increased heart rate, pupils dilate, digestion slows down, you get more alert, because obviously you've got to fight or flight or freeze from the danger. Once the danger's gone, the sympathetic branch is releasing noradrenaline, and that counteracts the effects of adrenaline. So all these will be kind of restored back to normal. This is sometimes called rest and digest. So this is a SAM pathway, sympatho-adreno-medullary pathway. And this helps to explain fight-flight response. Look at localization of function of the brain. So certain brain structures have certain functions. So obviously the motor cortex, which is in the frontal lobe, controls our movements and things. You can look at a picture of what's called the homunculus or motor homunculus, which is kind of a visual diagram of a slice of the motor cortex, and it shows you which part of that brain area is located for which movement, say. You've also got the somatosensory cortex in the parietal lobe. So this is a bit like the motor cortex, but it's somatosensory is dedicated mainly to sensory things like the face and hands, say, where you can feel things. The visual cortex is at the back in the occipital lobe. This is where we process visual information, and the visual information is what's called, it crosses over because it's contralateral, the brain is. So, for example, information coming from your left eye would get processed in the right hemisphere, and information in the left would be processed from the right eye would process in the left hemisphere. So it's contralateral. Auditory cortex is in the temporal lobe, dealing with auditory information, sound. So again, this part of the brain helps us to understand speech, sound. So Broca's area and Wernicke's area is linked with speech. So Broca's area is linked with speech production, and Wernicke's area is linked with speech comprehension, which are these language centers here. So you've got Broca's area in the left hemisphere, mainly responsible for speech production and language. So if you damage that part of the brain, you can end up with Broca's aphasia. And if you damage Wernicke's area, you could end up with Wernicke's aphasia. So if you've got Broca's aphasia, you'll end up with slow, effortful speech, maybe problems forming grammatically correct sentences. Whereas with Wernicke's area, if you had Wernicke's aphasia, you could have poor comprehension, lack of awareness of speech deficits. So depending on what part of the brain you damage, depends on the impact it has. So when we look at hemispheric lateralization, the brain has two hemispheres, which is connected by the corpus callosum. So the left hemisphere is well known for language, logical thinking, mathematic calculations, whereas the right hemisphere is more dominant for facial recognition, visual imagery, music ability, etc. So each hemisphere has different tasks, different structures, different functions. If the corpus callosum is cut, the brain is basically like having two separate people inside your head. So Sperry did a lot of split brain research, and information presented to one hemisphere could not be verbally reported if presented to the right hemisphere, because that lacks the language centers. If an image is presented to the left visual field, which is processed by the right hemisphere, patients couldn't name the object, but they could select it with their left hand because that's controlled to the right hemisphere. If an image is presented to the right visual field, which is processed by the left hemisphere, patients could name the object, but they might not recognize it with their left hand. So it's a bit like having two independent people in your head. But this does seem to suggest that the left hemisphere is typically used for language, and it demonstrates that each hemisphere can function independently and has different capabilities. It is suggested that consciousness might be divided into these patients, with each hemisphere having its own perceptions and responses. And it revealed that the right hemisphere has limited language abilities, but excels in spatial tasks. And it does show that the corpus callosum is crucial for integrating information. There are some good YouTube videos on Sperry's experiments with split brain patients. Now, just because you damage your brain doesn't mean it's damaged forever. You can get neural plasticity, which is more plastic when you're younger. Basically means that your brain can reorganize itself and form new neural connections. So, for example, in childhood, it's more supple, more plastic. If someone has brain trauma or stroke or disease, you can kind of regenerate the brain acts on, say, um, through rehabilitation, which is, physical rehabilitation, cognitive rehabilitation. So the brain can rewire itself and regenerate to a certain extent. And also, obviously, if the brain is used more for a specific skill, it can enlarge those areas. So, for example, there's a study by McGuire who looked at London cab drivers and found that the posterior hippocampus was bigger because they have to go through the memory process of learning the routes in London for 3 years. And then obviously you can recover certain functions, so functional recovery. So new connections could sprout. Other areas could be activated to kind of what's the word? Kind of allow a patient to have those abilities again and regenerate in a way. Denervation supersensitivity. So in terms of brain scans, fMRI, functional magnetic resonance imaging. So this is good for detecting changes in blood flow. Works on the idea that when the brain's in use, blood flow to that area increases to meet the demand for oxygen and nutrients. So if you have a functional MRI, that's a brain scan while you're doing a task. So this is how it works. Bit of a step-by-step process there. And then you've got the advantages and disadvantages of an MRI, an fMRI. So it's obviously non-invasive. It creates a nice 3D image, but you get poor temporal resolution. Temporal means time, so there's a bit of delay. But spatial resolution might be quite accurate in millimeters, which is this bit here. Okay, so in terms of how it works, so it uses a strong magnetic field. It aligns the hydrogen atoms in the brain. Radio frequency pulses temporality and disrupts that alignment. As the atoms then return to alignment, they emit signals that are detected by the scanner, and the active brain regions require more oxygen, therefore delivered by oxygenated hemoglobin, which is a protein in the blood. And oxygenated and deoxygenated hemoglobin have different magnetic properties, and the scanner detects those differences. So you get this bold signal, blood oxygen level dependent. And the computer then creates an image showing which brain areas are active. It's an expensive brain scan, but it is a very good detailed 3D image. It's got good spatial resolution. Some other scanning techniques, EEGs, electroencephalograms, or event-related potentials. So this is where you have like a swimming cap on your head at different deviations away from the scalp. Jasper in 1958 invented the 10-20 classification for this. And this can pick up brainwave activity. It's quite well known for sleep research. And this is how it works, which is basically what I just explained. And you get these different waves, alpha, beta, theta, and delta waves. Like I say, it does use a lot in sleep and also dreams. Whereas an event-related potential will be a specific response to something. So might be maybe moving a finger or something like that. You get a specific response that you're looking for. So an ERP, so a stimulus is presented multiple times while an EEG is recorded. The EEG segments following each stimuli are averaged together. Random brain activity cancels out, leaving only the response related to a stimuli. And the resulting waveform shows components that reflect different stages of processing. So because you get a lot of activity from anything you do, like blinking or moving or anything, a lot of the time you have to filter out the different bandwidths so you can only focus on the one that you're looking for. There's also post-mortems where people have passed away and you can study the brain in more detail. So if you're studying things like Alzheimer's or Parkinson's, the classic case of Phineas Gage, he was a railway worker. He had an iron bar that shot through his face and landed about 80 meters away. HM, that they provided insight into how brain damage can affect people's behavior. Transcranial magnetic stimulation. This is a bit like kind of Mickey Mouse ears that fire a pulse into the brain very, very quick, and it can disrupt the neurons. Uh, quite effective for depression, and it is a non-invasive procedure that basically uses a magnetic field to stimulate brain cells. PET scans or positron emission tomography. This is where you inject a radioactive tracer into the blood. There's a classic study by Tulving who looked at this. He looked at the difference between episodic and semantic memories, where they used PET scans. However, I think out of the 6 participants, 3 of the data was inconclusive, and Tulving was one of the participants as well. But this is obviously a little bit riskier for using radioactive tracers, but does give a good idea of different parts of the brain that are being used for different tasks. Biological rhythms, so sleep, wake cycles, circadian rhythms. So this is once every 24 hours. Now, in the new spec, you don't need to know this, but the circadian rhythm is governed by what's called the SCN, the suprachiasmatic nucleus, which is a bundle of neurons in the hypothalamus. It's basically the body clock, the master clock. This helps to regulate sleep-wake cycle. If you've ever been on a plane and you've got phase delay or advanced delay, your circadian rhythm might be out of sync. Infradian, menstrual cycles or hibernation, longer than every 24 hours. That's what they are. And ultradian rhythms are sleep stages. You tend to go through different stages. So you get REM, and then you get 90-minute cycles of these, maybe 3 or 4 times a night. So these are the sleep stages that people go through, less than 24 hours. But if you're doing AQA psychology, they've taken this out now, I think. The endogenous pacemakers and exogenous zeitgebers. So the endogenous pacemaker is your biological clock, which is governed by the suprachiasmatic nucleus in the hypothalamus. As mentioned before, the pineal gland produces melatonin, which is a sleep hormone, regulates sleep. So as melatonin goes up, obviously when it's dark at night, and then as the light hits your eyes in the morning, the melatonin will go down. Exogenous zeitgebers are anything that is like a cue. So clocks, meal times, temperature, social cues, they're all exogenous zeitgebers. So you know when it's time to go to bed, like when, say, the 10 o'clock news is on, or it starts getting dark outside, or a few hours after you've had your meal. They all kind of trigger you to know it's kind of bedtime. So you've got a bit of a better understanding now of some of the biological aspects in psychology, biopsychology. So thanks for watching, and I'll see you in another video.